to Likeable Science here on ThinkTech Hawaii. I'm your host, Ethan Allen, here on ThinkTech Hawaii, and in the studio today with me is Donn Viviani, a new postdoctoral fellow here at CMOR, the Center for Oceanic
Microbial Oceanography Research and Education.
Okay, and Elisha Wood-Charlson, the Research Communications person there at CMOR also. We use CMOR to CMORE because it's much easier than saying Center for Microbial Oceanography Research and Education, right. And we're going to be talking about microbes, molasses, and more today. So let's just jump right on into this. So why should sort of the man on the street care about the little phytoplankton and microbes in the ocean? That's
a great question. And to answer that, I'd like you to take 2 breaths with me. So, okay. So the oxygen for the first breath comes from terrestrial plants, so trees and shrubs and.
You can smell the pine.
Right, yeah. Exactly. But the second breath, the oxygen in the second breath actually comes from organisms that are growing in the ocean, so that are photosynthesizing in the ocean. And most of those are single-celled microbes because most of the ocean that's what makes up the majority of the things living in the ocean.
Okay, so it's just little tiny algae and plant-like things.
So if you care about breathing,
right.
You care about microbes.
There we go. They're important for us.
Right, right. And again, part of it is they're just doing their thing. It's not like they care about us. I mean, right. A lot of times we worry about microbes being bad, but the important thing to remember is that most microbes just don't care about you.
Right.
It's not they like you. It's not they hate you. They're just doing their thing. They're taking carbon dioxide and sunlight, and they're making new material and doing their thing.
Right, indeed. Even among bacteria, which we tend to think of as a bad group of microbes, something like 97% are either vital, helpful, or neutral to us, and only some tiny percentage, I gather, are particularly harmful.
Yeah.
So, but beyond sort of producing the oxygen that we breathe, these microbes also sort of are the base of the food chains in the ocean, right? So again, they have this huge vital role there.
Absolutely.
Yeah. Cool. And the other critical thing, of course, from all this, and as we're hearing about the climate change these days, is the role of these microbes in moving carbon around.
Yeah.
Right. So, and that's sort of what you've been studying, right?
Right. So, I've been looking at how fast these microbes are growing. So, if you think of photosynthetic growth in the ocean, you have this, this taking up of carbon dioxide and making more material, and then that material is then the carbon dioxide is in these microbes, and it can be used by the food chain. But there's actually a second part that I've been studying as well. So if you think of a plant on land, if you're a tree and you have plenty of nutrients and water and sunlight, you can add new leaves and bark and just keep adding cells. But if you're a microbe, there's a limit to how big you can get.
Right.
So either you get big enough and you divide in half and reproduce,
right.
But maybe you don't have all of the nutrients you need. And so a lot of times what microbes in the ocean will do is they'll release material because they don't have anywhere to store it and they're not ready to split. And so I've been looking at how fast the microbes grow versus how much of this dissolved stuff they release.
Oh, wow, they sort of spill their guts, as it were.
Except they stay intact.
They somehow know what to dump out.
Right,
it's not vital to them.
Yeah.
Interesting.
Yeah.
Cool.
And I like to think about that as being like a cup of tea. So if you think of a cup of tea, the stuff in the tea bag is like the microbial cells. It's a particle, you could pick it up, and the tea itself is the stuff that the microbes are releasing. Okay. It's a mixture of all kinds of proteins and sugars and so on.
Right. So there's a lot of carbon there that's now out loose in the ocean, as it were.
Exactly.
Which, as of course nature is very good about doing, nature picks it up and reincorporates it.
Exactly. It's that carbon is not accessible to bigger things like the things that will lead up to fish. But there are all these other bacteria that are living in the ocean that can take advantage of it. And so they take this dissolved material and they use it for energy and for building more biomass. And so that's how the carbon flows. Some of it is flowing to fish, but most of it is actually staying in this bacterial pool and just getting moved back and forth.
Okay, right. Getting into a bacteria may be eaten by a single-celled or small phytoplankton, small zooplankton,
exactly.
And then being released back out.
And the zooplankton are not, they're not particularly meat eaters.
Right.
So if you thought of trying to eat a jelly donut without your hands, that's
sounds like fun.
Right. That's what it's. I mean, that's how I like to think about it. There's a lot of sloppy feeding, and
Sure.
so you get all this material that gets spilled back in. And also viruses, when they attack these bacteria or these phytoplankton, will also release things into the ocean. So you have a lot of carbon is in the particulate, but an awful lot gets moved into this dissolved pool, like this tea.
Okay.
And so that's a major sort of source, or that's a major it's a major flow of carbon in the upper ocean.
Yeah, exactly. So we had a picture, I think, of this sort of the comparison of the land-based versus ocean-based.
Right. So this is so on the left-hand side under land plants, you see a seed leading to a tree because you're just adding cells, but then on the right for the phytoplankton, you see at the top you have this phytoplankton that has enough nutrients and energy to make 2 new phytoplankton. So it's reproducing, it's splitting, but the lower one doesn't. So it leads to 1 phytoplankton and then some dissolved molecules, and this can change, based on what the conditions are like. The same way that in the lower picture you see that trees can be either, they can have their leaves, or if they're deciduous, they'll lose their leaves in the winter in a temperate. Yeah.
So let me ask you then a sort of, maybe it's a naive question, but I mean, the classic, you show the seed going to the tree, and of course all that mass actually comes out of the air, right?
Yeah.
It's carbon that's pulled out of the air that's turned into these complex carbohydrates. And so presumably the same thing is happening with the phytoplankton. They're pulling carbon out of, in this case, out of the water.
Sure.
And turning it into biomass. And so there must be actually, again, just as with the tree, a surprisingly large amount of carbon being sort of sucked out of the atmosphere, out of the ocean in this case.
There is, but the thing to keep in mind is if you are an oak tree or a eucalyptus or whatever, you're going to live a long time.
Right, you're holding that carbon tight for a long while.
These phytoplankton in the ocean, they're getting eaten probably. Their lifespan is probably something like a day. So there's this really fast turnover, and that's because there's very little nutrient availability in most of the open ocean, and so the only way to keep all of these phytoplankton growing is to have this really rapid turnover where you grow, you get eaten, and that material is released, and other things take it up, and they grow and they get eaten. So you have this really fast turnover. Very little of that material sinks to the bottom of the ocean or moves up the food chain. Most of it is spinning very fast.
And it's good to remember on that figure that that's not to scale.
Right, that's absolutely not.
And so the tree obviously gets quite big and can produce a lot of cells that make up this entire tree. But the phytoplankton are single-celled organisms. They're microscopic. They're quite small. But when you think about the volume of the ocean and how many phytoplankton are in there, you can sort of see how you get the equivalent amount of oxygen production.
Right. I was going to say it must be the mass. The phytoplankton is probably actually larger than the mass of the land plants by a considerable factor, I would guess.
The biomass is, much bigger.
Yeah.
Yes. So that's how one breath comes out of the ocean from these little tiny single-celled things. There's just so many of them that they produce a massive amount of oxygen.
Yeah, absolutely. It is amazing the power of these microbes and the influences on our lives. And that actually brings us to a little aside I want to do. I do a segment here called Campfire Stories sometimes, and just interesting little, I think they're interesting little bits and pieces of science. And there are these microbes, and you guys both may know of these, that have, I say learned, but have figured out in some sense how to control animal hosts. So for instance, one species of them lives part of its life cycle in ants, but then needs to be eaten by cows. And it moves into the ant's brain basically and changes the ant's behavior so that the ant, rather than running around on the ground and avoiding high exposed places, actually climbs up stalks of grass, bites on the tip of the piece of grass and just hangs on there and then stays there, which does the ant no good at all, right? Except it puts him in perfect position to be chewed up by a cow, and these little beasts get into the cow where they need to be. And similarly, another species of them gets in mice or rats, who usually are very scared of cat urine for very good reasons, right? But this again changes their brain to where they find cat urine attractive and go sort of after it, which of course puts them in harm's way again. So microbes have an amazing sort of range of roles they play, things they can do that we wouldn't perhaps have thought.
So the rat-cat relationship is actually partly why pregnant women are asked not to change litter boxes is it's that same bug that potentially can infect them as well. Even though they want to be in a cat, they have these other hosts that they use as well.
Okay, and then it doesn't do the mom or presumably the child any good at
Yeah,
all.
it's so usually for adults it's not much of an issue. You can process it,
Right.
but if it does get the fetus infected, then it can have problems.
So it's somewhat parallel to the whole Zika
Yeah,
business, yeah. And interesting. I hadn't.
they're quite clever.
Yeah. And yeah, then they don't do well when they get in the wrong host, and they get unhappy and cause all kinds of
Well,
trouble.
things go wrong when
Right.
they're not in the right host. and
Right. Yeah.
that's-
Exactly.
We just tend to be in the way. I think, on that one.
Yes, we make these little bugs unhappy, and then they make us unhappy. That's the way it goes. But again, I think it does just talk to the sort of pervasiveness of little microbes and how much of the world we don't see, and until recently we've been quite unaware of. There are microbial populations now, they say, living kilometers under the seafloor even, I guess.
Yeah.
There are communities living, albeit I guess rather slowly,
Yeah.
but again, areas that we would never have thought before had any life at all. So it's amazing.
Yeah.
Well, that's a little bit of an aside, but we're going to take a quick break, and then we're going to come back and talk more about your work and about the microbes in the ocean.
Great.
We're talking about microbes, molasses, and more. I'm your host, Ethan Allen. Donn Viviani and Elisha Wood-Charlson are with me here from CMOR, the Center for Microbial Oceanography, Research, and Evaluation.
Education.
Sorry. We'll be right back.
Aloha, my name is Richard Emery, and I host Condo Insider. We talk about issues facing the Condo Association throughout Hawaii and talk about solutions. When you think about it, about 1/3 of our population lives in some form of common interest real estate. We broadcast every Thursday at 3 p.m. Please tune in, and thank you. Aloha.
Hi, I'm Donna Blanchard. I'm the host of Center Stage here on ThinkTech. This show is so very dear to my heart. We talk with artists of various different ilk here about the process that they go through for their art. So we talk about what they're doing, why they are doing it, how they do it. And it's a show that is inspiring. This is what I hear from people all the time. And a show that will teach you something, sometimes something about yourself. I hope you'll join us. The show is Center Stage. It's on ThinkTech every Wednesday at 2 o'clock. We'll see you then.
And you're back here on Likeable Science. I'm your host, Ethan Allen, and with me today are two folks from UH-CMOR, the Center for Microbial Oceanography, Research and Education. I got it right this time. Elisha Wood-Charlson and Donn Viviani. Donn's a recent PhD out there, and did it like a week ago. Congratulations.
Thank you.
And he has done pretty amazing research out on the microbes of the ocean here. So, what you sort of talked about going out there, looking at the dissolved organic carbon and looking at some microbes. Sort of, how do you do this? What does this involve?
So I'm really fortunate because we at UH have the Hawaii Ocean Time Series that's been going out 60 miles north of Oahu for since 1988 and has been measuring the physics and the biology and the chemistry, the biogeochemistry of the ocean at basically almost every month. And so that gives us this really nice time series of what happens. And so it's a really nice place for a graduate student to go along and go out to sea for 3 or 4 years and make these measurements and then put them in the context of, okay, we know that over the course of this time series, we know that in the summer, the photosynthetic organisms, their growth increases and it decreases in the fall. And knowing that, you can start to ask questions. You can say, okay, so as I did, I said, all right, we know that there's this increase in the spring and then it's higher in the summer. Does that hold for the production of this dissolved organic carbon? And so that was, right. And it turns out that when we went out and measured it, it didn't. We didn't have this seasonal pattern in the dissolved organic carbon, which suggests that there's something different controlling the production of the particulate and the production of the dissolved. So the tea doesn't match the tea bags.
Right. Okay.
Yeah.
That's intriguing. We'll probably get into later on as to
Okay.
why that might
Sure.
be.
Sure.
But there is a rhythmicity of it, right?
Sure. So I did some experiments looking at, okay, so what happens when you look over the course of a morning and compare that to the full day? And the production, the growth, the photosynthetic growth of these phytoplankton doesn't seem to change between the morning and the full day. So it's sort of a steady rate of production. But the production of this dissolved organic material is higher in the morning than it is over the whole day. And it may be that, I mean, this is opinion, this is speculation here.
Sure.
But, it could be that, when the sun comes up and the machinery turns on, they're just trying to build up some energy stores. And so they're just making simple sugars and releasing them just to get the energy. It could be that when the phytoplankton start growing and they start releasing this carbon, the bacteria that are going to take it up haven't caught up yet. And so it takes them a little while to start taking all that stuff. And so you have this increase in the dissolved, and that starts to come down as the bacteria start to grow up. So it could be those things.
Sure. Yeah, these ecosystems are very complex interactions among many different life forms, all using the same kinds of resources, produce things that are used by others, and
Right, and possibly offset in time, or there's some signal that one group gives another group.
Right.
So these are all things that we're looking at.
Excellent, excellent. And so now bacteria are part of these same ecosystem, and so do the bacteria have the same pattern?
Sure, that's a great question. They do. They have a pattern. When you look at them over the course of a day, again, you have this sort of increase in the morning of this dissolved organic material that the bacteria are using. But the bacteria, the growth rates of these bacteria don't actually peak. They don't hit their peak until about sunset. So they're offset from the production by hours, and it may be that it's a temperature thing. It could be that over the course of the day the temperature increases and the bacteria, their metabolism are just tuned to temperature, or it could be that the right kinds of materials just aren't available for them until later in the day. It could be either of those things, or other things.
Sure. Okay, and that's an interesting disjunction of these various cycles, but ultimately it all works out, right? That's the beauty of the ocean.
Yeah.
But again, they don't show much of a seasonal signal either. The bacteria.
No, they show. I have less data for them, but what I do have, it looks like that you see this increase in the photosynthetic growth in the summers. And the bacteria their peak in growth is later in the year. Okay. And again, that could be temperature related because the sun day length increases throughout the summer and then starts to decrease. Right. But the temperature of the ocean continues to go up into the fall. Right. So it could be that it's a temperature thing, or it could be different types of bacteria growing up over time type of thing.
Yeah, and of course, so many of the biochemical processes in bacteria, but in other things too, are very temperature dependent, right?
Absolutely.
And, these reactions will go faster or slower depending upon.
Exactly.
Yeah. So, but what is it? What's it like to go out there? I mean, you said you said 3 to 4 years. A lot of small 4 years is not one long.
Right. Yeah. So there's been 270 hot cruises.
We're on like 282.
Oh wow. Okay. So it's they're 5-day cruises. So we and at this point they're the technicians and the scientists who go out on these cruises are very, very excellent. They work really well, but it's practiced. It's like going to work. I need my lunch. I need my bus pass. I need it's the same thing with these. You go out. You leave in the morning. We get to Station Aloha, which is our study site. In the evening, we put instruments in the water. We send down instruments that measure the temperature and oxygen and chlorophyll and density of the water to get an idea of what the water column is like. We work 12-hour shifts around the clock just to sort of take advantage of being out there. The measurements that I made, we would take bottles and fill them with seawater.
Right. We've got, I think, pictures of those, if I'm not mistaken.
This is from when we went out and sampled the molasses patch.
Okay.
Which was a similar sort of undertaking, except that with this, when we go on a hot cruise, we know exactly what we need. We have a list of the things we're going to take, and there's a checklist. With this, we didn't actually know what we were going to need.
Okay.
So we sort of filled the boat with everything we could. So it was a sort of a tight fit. But here we're lowering a smaller version of the kind of bottle that we use to collect water from depth. It's a Niskin bottle that will close and trap the water inside, and then we can bring it up and we can open it up and get the water at the conditions that it was at when we collected it.
Excellent.
One of the great things about being at sea is that if you sort of like start looking at time lapses of people doing this, they're basically putting all this instrumentation in and leaving it for a little while, and it sort of floats around. But basically, they're just pulling water samples out. And so you're in a particular area at a particular time, and you need to try and sample as much of as possible at these different depths across the space. And so it's pretty much just a 24-hour, round-the-clock sampling of water. So bottles go in, bottles come out. Those get sampled, bottles go back in. And so all of that comes back, and then Donn does a lot of his work in the laboratory to then process what's in that water, what has that water been doing.
Sure. So you've got to, of course, have a very good systematic approach. Again, this is the essence of science, is to be very systematic about how you do this, and you know which bottles were which. It wasn't just, one of these bottles.
Yeah, there's, I mean, they measure so many different parameters when they're out there in terms of like nutrients and salinity and all of that. So when you come back, you have a bottle that's been nicely collected, and it has environmental context that is around it. So you can actually take a particular bacteria out of it and say, what was that bacteria experiencing at that time?
Right, because sort of in concert with that bottle, which you know was collected at a certain depth at a certain time, you know what the temperature was in that nearby water, you know what the salinity was, you know a whole bunch of the pH, maybe the currents, whatever it may be. You know.
And the light, so you can kind of get an idea about, you know, if it's cloudy versus if it's full sun, like how does that affect, for example, carbon production?
Right, so yeah, you can look at a lot of different, you can ask a lot of different questions then. Yeah.
And like I said earlier, it's a great opportunity for somebody to come in and ask a specific question, because you can sort of look at all the years of data and say, here's a pattern that we see. How does this one thing affect it? Or what happens if we measure this other thing along with it?
Right, and more there are great tools out there now, I gather, to begin to look at these big data sets. And when you get these multiple data sets now, it's harder and harder to really comb through all this stuff by hand, right? I mean, they're massive numbers of massive numbers. And,
so that's where the bioinformaticians and programmers come in handy. So computers sort of sort all that out now, thankfully.
And of course, gets to be a sort of a tricky business, because if you have enough data sets with enough numbers in them, you can get spurious correlations, and that mean absolutely nothing other than, oops, I had enough numbers.
Well, and I mean, the complexity of the ocean, every time we go back in it, is more and more. And so, the correlations might be correlations, but there's one pivotal feature that you didn't expect. So, for example, there was a recent publication that came out of Ed DeLong's lab that Jessica Bryant did. And she looked over, years of this time series data and tried to figure out what some of the patterns in the bacteria were, and ended up coming up with wind. That seemed to be the one thing that actually explained a lot of the variability. It was not necessarily anything anybody expected, but because you have all this contextual data from 1988 all the way through, it provides this really, really resource-rich place to ask these really interesting questions, and then you come up with wind. It was actually what happened. So questions that we never would have been able to answer without long-term time series.
No, that's the real value of that kind of good stuff. Excellent. I want to take another little sort of side step here and talk about microbes in a slightly different context. It actually harks back to a guest I had on the show here about a year ago, a gentleman named James Smith from the University of Virginia, who talked about his new point-of-use water decontamination system called Maddie Drops. And these are tablets that he developed that are ceramic tablets that are mixed initially with sawdust and with some silver nitrate and then baked. And of course, in the baking process in a kiln, all the sawdust burns out, leaving a very, very porous ceramic that also is infused now with silver. And when you drop these into water, a container of water, over a period of hours, a tiny bit of silver ions are released at what seems like a fairly modest concentration, far below anything harmful to people, but quite lethal to almost every microbe. Viruses, bacteria, fungi, all kinds of things are very sensitive to silver. And so after a few hours, your water, even if it was really quite undrinkable before in terms of very badly contaminated, is perfectly drinkable. And these finally have actually just in the last couple of months become available now. And I just sent some out to my study sites out in Micronesia, which is going through a severe drought, which means people are, of course, drinking water they should not be drinking. And I'm hopeful this will be a sort of a game changer technology. But again, it's microbes playing another role here, right? Another place where microbes intersect with our lives in ways we might not expect, right? So we are going to jump out in another moment or two, but when we come back, we're going to talk about the thing we sort of previewed there, the molasses spill, which was a great sort of natural or unnatural experiment in a sense.
Right.
But we'll talk about again how something like that ties into the microbial populations and the issues that you're studying 1,000 miles away, right? 600 miles away, okay.
60.
60 miles, oh, okay, sorry. But we're gonna do that when we come back. Right now, I think we're gonna take another break. And I'm Ethan Allen, Donn Viviani and Elisha Wood-Charlson of Seymour are both with me here today in ThinkTech Studios, and we'll be back in a moment.
Hi everybody, Ted Ralston here, your host on Where the Road Leads on ThinkTech Hawaii, where we talk about the intersection between technology and the quality of life here in Hawaii, where we're always exploring where technology can come into the game to help everybody enjoy a better and a higher quality of life. We bring guests on, such as my guest today, Brendan Brennan. Brendan, thanks for coming on.
Yeah.
And
Aloha, namaskar, and hello. My name is Anu Hitchell, and I host the show called Climate Change Beyond Outrage. We go beyond outrage to find solutions to climate problems facing people, nations, and the world. I hope you will join me here every Tuesday at 1 o'clock. We broadcast live from thinktechhawaii.com. Aloha and bye-bye.
And you're back here on Likeable Science. I'm your host, Ethan Allen, here on ThinkTech Hawaii. With me today in the ThinkTech Studios are Elisha Wood-Charlson and Donn Viviani, both from the Center for Microbial Oceanography Research and Education, Seymour. Hey, I got it right this time. It's one of the real uses for a good acronym. We've been talking about the microbes that Donn studies, and particularly microbes that live near Station Aloha, sort of out in the middle of the ocean, 60 miles north of Oahu here. But one of the beauties of science, of course, is science often presents us with sort of strange opportunities. And you had one of these a little bit ago, right?
Yeah. I was one evening, my advisor, Matt Church, called me and he said, have you been watching the news? And I said, no. And he said, there's this enormous molasses spill. We've got to get out there and take a look at it. So he'd been talking with some other folks at Seymour, and there was this, in 2013 in September, there was this 260-some thousand gallons of molasses spilled into the Honolulu
big
harbor.
pipe rupture from
Right.
right.
Yeah, and the pipe was broken sometime over the weekend or Monday morning, and it wasn't shut off. It wasn't noticed until Monday morning. It wasn't shut off until later, and so you had this enormous spill of molasses. And I made the point earlier that you have all of these bacteria that make a living on dissolved organic material, and molasses is just dissolved. It's dissolved sugar with some nutrients in it. So you had this enormous amount of molasses in the harbor that was visible, and you had all these fish dying. And so we, as I said, we a bunch of us said we should go out here and look at this. You could never get permission to do an experiment where you dumped, hundreds of thousands of gallons into a harbor.
Right.
Yes. But when you have this opportunity, we decided to take advantage of it. So we piled everything we could fit into the boat in the boat. To move, we were basically walking around the outside of the boat because the inside was full of boxes of bottles. And we sampled along. We tried to find a place in the harbor that was outside of where the molasses was, and then we moved closer to the source. And we got out there 3 days after the spill, and it was amazing to see sort of the difference. I mean, there was, at our site away from the spill, it was part of the problem is we don't have a good idea of what the harbor looked like before because we don't have a baseline. Nobody had gone out and sampled it, but at the space outside where the spill was, there was plenty of oxygen in the water. There were no odd smells. But as we moved closer to where the spill was, the amount of oxygen in the water went down to basically nothing. We had this instrument that we could lower and get a concentration of oxygen, and there was basically no oxygen at some of these sites closer to where the spill was. And at one of the sites, we actually smelled this, sort of rotten egg hydrogen sulfide smell that suggested that you had sort of different types of microbial growth happening there.
That's right, because that would be from like some bacteria that usually are a tiny component of the population and don't do very well in normal conditions. But now suddenly
you have this no oxygen water column.
And the reason for the no oxygen was because essentially
so normally you have this balance between the algae or the bacteria that are like algae that are growing on CO2 and sunlight, and the bacteria that are growing on dissolved organic stuff. Well, when you add all this molasses, there's all this sugar, and so the bacteria were taking this up, they were consuming it, and they were breathing,
Growing, breeding,
dividing, and as that was happening, they were using oxygen, and they were using oxygen so fast that the photosynthetic algae and oxygen coming in from the air couldn't keep up. So they basically just breathed all the oxygen down, and at that point, you know, it was not a great environment for fish and other things because they were unable to breathe, and probably bad for a lot of the bacteria. And so you start to get other types of bacteria that, as you said, are normally at very low concentrations, but could make a good living in this anoxic environment.
Right. Yeah, that's a nice example of the dynamic nature of the ocean, and hopefully would inform the study. I got these dead zones, not from molasses, but from other things, are becoming increasingly frequent now. I know they've had some bad ones last summer off of the Pacific Northwest coast, and of course there's almost a regularly recurring one at the mouth of the Mississippi now every year.
So you'd mentioned how, you know, things just work in the oceans, and that's sort of where Donn did most of his PhD, where you can study how much photosynthesis is going on. Photosynthesis obviously produces oxygen, and then you can study, you know, the microbes that are eating all of the carbon that has been fixed by photosynthesis, and then they are like us, they respire carbon dioxide and use oxygen. And so this was in a system where they basically just added, I think the analogy was honey to the tea.
Right.
And so all of these other microbes that use oxygen and the sugar and release carbon dioxide basically pulled all the oxygen out of the environment. And things are sort of happening in the dead zones where you get a balance that's been shifted.
Right.
And the oxygen just can't replace itself.
Exactly. People don't realize how fast microbes reproduce and the sort of the geometry of this. The classic example is that if you took a single paramecium and allowed it to divide every 20 minutes, which they'll do under ideal conditions, and could feed it enough that in 24 hours you would have a mass of paramecium, I believe, equivalent to the mass of the Earth, in theory. Nothing much happens till, you know, about hour 22, but you know, suddenly it really begins to grow very rapidly.
So when you do that with molasses, that's exactly what happens until something like oxygen becomes limiting. And then the whole system crashes out, and then the ones that can survive without oxygen sort of finish off what's left in terms of the carbon, and that's
gradually,
the smell that, that you get. So.
Excellent. Well, that's a beautiful example of how sort of science can run, and how scientists actually have to do some of their work, and how sort of your training actually really puts you in a mindset to say, hey, you know, this isn't just a disaster. This is oddly sort of silver lining in a way for us. It's silver lining for the bacteria, clearly.
Well, to a certain point. I mean, it was silver lining. It was good for them on the short term, but you know, when we went back a week later, you know, conditions were more or less back to normal. So you sort of had a restoration.
Sure.
I mean,
hysteresis and all.
Exactly. We noticed at our second site that was starting to get into where the plume had been, there were all of these tiny but visible organisms that were grazers of bacteria, and so they, I'm sure, had a field day eating all of the molasses-fed bacteria. And so, you know, the bacteria shot up, but over time they were probably controlled, and you know, mixing would bring oxygen back in, and so the system sort of restored itself, given a little bit of time.
Do you think there have been real long-term measurable impacts in the harbor from that, or has it pretty well probably come back?
I mean, I suspect that I think people have been looking at the coral, and I think that I think it was rough on the coral. That was.
Unlike bacteria, they live for a very long time, and they reproduce very slowly, and so those sort of life history strategies will take a little bit longer to recover. But in the sense that, you know, ecosystems tend to be fairly resilient. If it's a one-off, short-term, acute disturbance, then they can kind of recover from it.
Right, but the coral, of course, are facing actually multiple threats of
they've got some
warmer water, more acidic water, other pollutants besides the molasses.
There's some multipliers on the coral health, unfortunately.
That's that is unfortunate, but and again, the coral are a very interesting part of this whole ecosystem too, with their own microbial components, basically.
Really interesting symbiosis.
Yeah.
But you know, we did see. I mean, even on a week later, we saw, you know, fish coming back in, and so, you know, you could see that the organisms that could move were moving. It's just the ones that sort of rely on random dispersal are going to take longer.
Right. I guess I've seen some of the photos of the sea floor and some of these dead zones, and the crabs, we can't move too fast sometimes, end up littering the floor, basically.
Right. Which I think was, people were taking pictures of here in the
Oh, okay,
molasses
okay.
patch.
Interesting. So, talk a little bit about what got you into this whole business. What keeps you sort of interested in, and how did this sort of start for you?
I mean, I used to be I used to teach science in a classroom. I taught middle school science, and I enjoyed teaching, but I also I realized I was sort of thinking about I would be teaching science. I'd be thinking about questions that I would be asking about things that I was teaching, and I started thinking about oxygen. Actually, it was dissolved oxygen in the Pacific Northwest, and I ended up doing a little bit of work there, and so that got me into measuring oxygen concentrations and changes, and that got me into measuring respiration and production of oxygen by photosynthesis out here in the Pacific, and that's sort of what got me interested in. And I think having these sort of these changes in time and trying to figure out how things line up is the thing that keeps me going. I find it really interesting.
Yeah. Science typically, it does seem it really engages you because it's not ever quite the same.
Yeah.
You may be doing repeated measurements, but either the system is changing or the way you're measuring it may change or whatever. Yeah.
Yeah, and it's going to sea, doing field work is fun, and it sort of keeps you on your toes. You can go out one January and the seas are really high and the winds are really strong, and it's a struggle to get instruments in and out of the water, and then you go out another January and it's flat as a lake, and you can see Oahu from 60 miles away. It just kind of gives you it keeps you on your toes, and it keeps you interested.
Yeah, excellent. And obviously, as a graduate student, you've been through sort of a long educational process here, just completed a long educational process, and congratulations again.
Yeah.
So what advice do you have for sort of aspiring scientists based on your experiences here? Run.
No pressure. No pressure. I might jump in a little bit
Sure,
and just say, I mean, I think you brought up a good point. Like, you were teaching science and kept asking questions that weren't necessarily answered in the book. And I think anybody that wants to know a little bit more isn't quite satisfied with what they're learning in class, like, has a natural tendency to ask the right questions. And I think those people should definitely pursue those questions. And that's really how I got into science as well. I was like, well, I want to know a little bit more about how this works. And then you end up realizing that there isn't really an answer, and you go find it yourself. And then you end up with a PhD, and you're like, okay, here I am. And so I do think that anybody that's interested in how the world works, and especially now with things changing so quickly, and really if you feel some compassion for wanting to understand how it's changing and how we're going to adapt to it, science is definitely where you should be.
Yeah, absolutely. That our ability, because we have both a larger knowledge base now, and we have these just rapidly increasingly sophisticated tools.
The technology is amazing.
Yeah.
On every front, from studying the brain, studying the ocean, to studying rocks, I'm sure. We can find out so much more from so much less now. I mean, I gather it's become a real issue, the classic turnaround in a short generation time, DNA analysis. It's become to the point where they can measure DNA off of such small samples that you can get sort of residual, sort of secondhand DNA contaminating, say, a crime scene, and it gets very confusing sometimes about whose was this, how did it get here, how much of it was there. Yeah, so it's a whole new world out there.
Well, and it's moving so fast that, if you do like gadgets and you're into technology and stuff like that, like, to be honest, scientists get to play with most of the advanced technology and part of it, but the questions always get more complicated, and we always need more tech. So anybody that's into gadgets and stuff like that and has ideas on how to build it, like, we need you in science.
Yeah, absolutely. It's a great place for I was interviewing Song Choi, your Associate Dean of Engineering, and he was talking about engineering as a creative endeavor, and he said it's the people who really want to do something who come into engineering. I had never engineering doesn't have that particular vibe to it really, but he's quite right in that sense. And yeah, you put it very well there. That
And we partner with engineers all of the time
Sure.
because our questions push instruments, especially in the ocean and
Sure.
out
Yeah.
at sea in these crazy conditions.
you're saying, hey, how
push
can I
it to the
measure
limit.
this?
Right.
You know, and,
Yeah.
and it's got to be this small and weigh only
Yeah.
this much.
Or going out on a monthly hot cruise is an enormous investment of time and resources
Right.
and so on. And it's important to do because there are some measurements that you need to make while you're there. But if we can sort of combine that with floats and with gliders and satellites and these other things and tie those together, we can get an idea of what's happening in time between the times that we're able to get out to sea.
Exactly.
And a lot of the technologies are getting cheaper and smaller. I mean, the CTD that we use, the instrument that we send down with the bottles to measure the temperature and the density of water and so on. There's actually an open source Arduino kit that I think you can use to put together a basic CTD that in your own garage so there's a lot of opportunities for people to sort of get into this even as a hobby or.
Sure. No, it's amazing. One of my other past guests on the show is a guy at Stanford who developed the 50-cent microscope, the fold scopes.
Yeah.
Yeah, just an amazing. Like, wow, how do you bring the technology back to that simple level? Amazing stuff. Well, this has been excellent. This has been a very informative discussion. I feel like I've learned a lot, and I've enjoyed it too. It's always fun talking with you, Elisha, and very nice to have met you. And we've been talking about microbes and molasses and all kinds of good stuff. And I wish you the best of luck then in your future endeavors. And we'll hope that we'll see you both again sometime soon. This is Ethan Allen here, your host on Likeable Science, signing off for another week. Hope to see you next week.